Welcome to our seminar series, kicking it off for the fall quarter. It's my tremendous pleasure to welcome Jessica Metcalf here. Jess completed her PhD at Imperial College, working on evolutionary demography of monocarpic perennials, just like her title. And then postdoc work on the evolution of senescence at the Max Planck Institute of Demographic Research, and then postdoc work on tree demography at Duke University. Then she saw the light and got into infectious disease dynamics. Came to a postdoc at Penn State, which is where I got to meet her because we were fellow postdocs there for a while. Then went on to hold a Royal Society Fellowship at Oxford, and then joined the faculty at Princeton in 2014. And she's been a joint appointment there in EEB and public affairs, interestingly, and has just been made full professor at Princeton. So Jess does, I would say, very elegant and insightful modeling on a broad range of problems, from fundamental issues in the population biology and infectious disease through to very applied issues like advising the WHO on vaccination policies and how to eliminate pathogens for countries and so on. And through it all, she has a consistent track record of carrying ideas and tools. You can have it, sorry. From one discipline to another. So from the worlds of demography and evolutionary ecology into infectious disease dynamics, we benefited from that. She spearheaded the introduction of integral projection models, IPMs, into our benighted world. And she's also been a leader in thinking about how we can improve our understanding of the risks of pandemics and epidemics by better characterizing the landscapes of immunity out there in human populations. And I think that's what we're going to hear about today. So please join me in welcoming Jess.
Well, thank you so much, Jamie, for that rather over-the-top introduction. And thank you, everyone, for being here. So the estimated 10 to the power 33 viruses that constitute the planet's virome are arguably the most successful self-replicating entity on the planet. Every progeny virus, on average, has one mutation, and an infected human can produce 10 to the power 12 virions. So if you're an evolutionary biologist, it seems clear that in evolutionary terms, we are in a very unequal battle. My very favorite definition of a virus is by Medawar, who described a virus as a piece of bad news wrapped in a protein. And as you will perhaps remember, in 2020, the news was very bad indeed. From very early during the pandemic, we knew an incredible amount about the pandemic virus. People at all career stages and from all parts of the world worked unbelievably hard to make this happen. We knew things about the R0, we knew things about the serial interval. I'll bet people in this department contributed to that in ways that really were spectacular. And I think were, the grit and sweat of numerous people were just remarkable contributions. But the dust has settled a little bit, and so it is perhaps time to step back and think about the bigger picture and think about emergent pathogens in general. What I'm going to do today is take that perspective and ask three different questions. So think about why there are only 4 endemic coronaviruses, why MERS-CoV in some sense failed. So this is a different emergent coronavirus, whereas SARS-CoV-2 succeeded, not from our perspective, obviously. And then a little bit about landscapes of immunity. So to start with the first question, emergent pathogens are nothing new. So pathogens have emerged and re-emerged on the planet. This is just showing numbers of cases on a log scale, number of recorded cases on a log scale for the last 20 years. And SARS-CoV-2 should actually be way off the graph in this plot now, because this was the right height when that graph was made. So to me, disease emergence is not surprising per se, but if you look more closely at this, what you'll notice is that over the last 20 years, we have had 3 different coronaviruses pop up into our populations. SARS-CoV-1, then called SARS-CoV, because to call it SARS-CoV-1 would have been the act of a pessimist, I think. MERS-CoV, so Middle Eastern Respiratory Syndrome, and then SARS-CoV-2. And this is peculiar, as a brilliant postdoc in the lab pointed out, because we do have, as Benny Rice noted, 4 endemic coronaviruses, 4 and only 4 endemic coronaviruses. They have completely unmemorable names like 229E, et cetera, but even if the same rates of spillover had happened over the last 100 years, we shouldn't just have 4, we should have, like maybe 15 or so. So why are there only 4 endemic coronaviruses? There are their names in case you wish to lighten them. And of course, the first obvious possibility for why there are only 4 endemic coronaviruses is that there are not, in fact, only 4 endemic coronaviruses. We haven't counted right. There might be more endemic coronaviruses than we think. So what we know is that the what's called the viral discovery curve has been flattening. This is a paper from a few years ago, but it does seem reasonable to assume we have a pretty good handle on the numbers of viruses that are circulating within our populations. It's not entirely robust, but it's not without possibility. The viral discovery curves flattened, and furthermore, if you were to make an error in counting, you'd think you'd be more likely to underestimate these rare emergents that might flash up and then disappear the way MERS-CoV does, rather than these endemic things that are circulating continuously. So the chances are that we can probably reasonably assume that we've counted right, and the number is about 4. Another question might be, has the pace of spillover accelerated? It feels very strange saying this with Jamie in the room because he knows much more about this than I do. I'll keep going. So if the pace of spillover has accelerated, that might be another way in which we've had these 3 emergents in the last 20 years, whereas we only have 4 endemic coronaviruses. And there are many ways in which this might happen. So people have suggested that the intensity of anthropogenic disruptions to natural systems has changed. And this is certainly true, but it's also true that zoonoses go back hundreds of years, from the 1918 flu to things like the plague of Athens. They aren't something entirely novel. So this isn't impossible as part of the explanation, but it doesn't seem like it might be the whole explanation, especially since some of these examples predate industrial extractive processes. Of course, another angle is that travel and connectivity has vastly changed. So this is flight connections in 1933, and it'll come as no surprise that in 2020, this is flight connections. And of course, this is great if you're a virus emerging because you can go everywhere very, very effectively, very quickly. But again, here, like the 1918 flu did fine, even before the 1930 connection. So this might be part of it. There's also evidence that some sort of coronavirus seems to have bubbled through onto the world stage in the late 1800s. So it might not, again, be the whole story. I think a really interesting piece is that we ourselves are changing, and we're changing in that we're getting older, something I feel every day. And this might be, we know very well that senescence is very much part of, unfortunately, part of life. And this is something that manifests in immunity. So our ability to keep infection, novel spillover pathogens out of us might decline as we are older, and it might be reduced in populations that are predominantly elderly. On the other hand, you can imagine that the people who are going out and adventurously coming into contact with bats or climbing through jungles might tend to be on the younger side. So that the degree to which this plays out as a role is not quite clear. One of the other threads is, of course, maybe there are limits to pathogen biology. Maybe only a very limited number of pathogens have the right set of ingredients that will allow them to latch onto our cells, to get into our cells, to replicate within our cells. And the first piece of that story is that they have to have the right degree of receptor binding. So they usually bind onto a receptor on our cells and then kind of wiggle in, etc. And for SARS-CoV-2, famously, this is the ACE2 receptor. But it's usually some sort of, you can think of it as some sort of lock and key mechanism, and they're going to need the right key to get into the lock of our cells. And maybe it's just vanishingly rare to get that right combination. The evidence against this is, first of all, the coronaviruses are incredibly diverse. There are thousands of species in avian and mammalian hosts. I mean, in a way, this makes the question kind of like, why do we only have 4? If we were a pig, we'd have thousands. If we were pigs, perhaps not aping. So it doesn't seem like barriers in receptor ecology are likely to be the full story here. And if you look, at the ones that we've had, so this is a phylogeny of coronaviruses across the tree of life, and there are different hosts sort of plotted on there. And it does seem like the ones that we have, so the ones shown in red and in orange, yellow, come from all sorts of different host species. So it doesn't seem like they're like really struggling to find a way into our cells particularly. They're coming from coronaviruses that have been separated for vast amounts of time are still equally able, it seems, to come into our populations. Those that do come in are all using different receptors. So SARS-CoV and SARS-CoV-2 use the ACE2 receptor. That Middle Eastern respiratory syndrome uses a DPP4 receptor, and the endemic ones use a mixture of different things as well. So this lock and key thing seems like they can be pretty good at using different keys to get in, different locks and different keys. And furthermore, we know that there's been a ton of spillback. So that you may remember back early in the pandemic, gorillas and zoos were getting infected with SARS-CoV-2. Ferrets, famously, and mink in mink farms. And then, of course, deer all over Pennsylvania seem to have coronavirus. So it doesn't seem to have, it's not particularly held back by its receptor biology, at least across hosts. Another thread that might be an important barrier to how coronaviruses get into us is that there might be limits more around the pathogen biology. And just a possibly unwelcome reminder of how this works. This virus binds the ACE2 receptor, enters cells in the respiratory tract, starts to replicate, and in the right, one is hoping that it will be cleared by immunity and we can move on with our lives. If it moves down deeper into the lung, you get worse symptoms. And I mean, just as a reminder, the lung is a single cell wall thick, so nothing can really go wrong in the lung. It's a very delicate place from the perspective of a virus as well, right? It might be really hard to be a virus that effectively replicates within a host without killing the host too quickly. If you kill your host too quickly, you're not transmitting. This also might be a very hard needle for the virus to thread. So keeping your host alive, if you're something that inhabits the lungs, might be difficult. Host immunity is, of course, one of our main lines of defense, and we have two flavors of immunity. We have innate immunity, and we have adaptive immunity. And innate immunity is just the pieces that go looking for parts of pathogens that are highly conserved. And it does seem to be the case that quite often pathogens evolve to look more and more like our cells, so they're less likely to be detected by, for example, innate immunity. So it could be that, it's quite hard to get through those types of defenses for coronaviruses. There's plenty of evidence, of course, that some pathogens get lucky and seem to slip through these barriers. We did have suggestions even from early in the pandemic that adaptive immunity was limiting. So these are the 3 endemic coronaviruses. And this was, I think, a time series from Scotland. And what you see, this is over time, is that the abundance of the orange guy tends to be big when the purple guy is small and vice versa. And this suggests that the thing that our immune system is learning about the one is allowing it to repress the other to some degree, possibly. So if there was no overlap, if the non-overlap within the time series is at least suggestive of patterns of cross-protection. So if you've seen one, you're protected against another. And this might lead to some sort of landscape of immunity where, you can imagine there's these 4 endemic coronaviruses, and where they sit, what they look like, how they sit in this kind of phenotypic space, creates a bubble that are of, people who are protected, who have seen this landscape. So the only places a new thing can come in would be kind of sneaking into the corners, into the dark green areas. Jamie has done lovely work on monkeypox, for example, that has spilled over leveraging, Brian word, using the fact that smallpox vaccination had diminished. So suddenly there was no immunity to smallpox in the populations, which meant that monkeypox could more effectively infect people. So it might be a similar sort of pattern here. So all of this is speculative, but I think it gives us, ways of thinking about the big picture of how these sorts of pieces can come in by thinking about a combination of what is endemic and what is emergent. And the storyline here is that 4 endemic coronaviruses does seem like a small number. And if we think it's not reporting biases and not too much change in recent conditions, then it's likely that there's something to do, or we can bound the number of things, the number of explanatory variables that must be in play. Transmission, virulence, and existing immune cross-protection. Suggesting overall that to successfully emerge a new species of pathogen to come into our populations has to be able to thread this needle in terms of being just the right degree, virulent but not too virulent perhaps, or not virulent at all is even better, of course, but then we wouldn't care. Transmitting enough and evading existing immunity within human populations. The coronaviruses give us this one tool for thinking about pathogen emergence, but it also gives us another window onto this question, and that is that while SARS-CoV-2 was so rampantly successful across human populations, Middle Eastern respiratory syndrome seems to have come over quite frequently and yet not gone anywhere, which is a bit surprising, right? This is an infection which is a very mild infection in camels, and there's really high seroprevalence in adult camels, suggesting that it's transmitting very happily and very rapidly in camels. Possibly it's adapting to camels. There does seem to be a suggestion that the most recent common ancestor is not so long ago. But the reason that we're concerned about it is that more than 2,000 cases have been recorded within human populations. And these originate from multiple spillover events. So it comes over seemingly quite regularly. And you get short chains of onward transmission within households. And then occasionally, like there was a case in Singapore, Hong Kong, Korea, where there was more than I think there was hundreds of cases that happened in a nosocomial outbreak in a hospital. Okay. And I remember in 2014, there was one of these little outbreaks. It had come over from a camel. It had spread there'd been a few chains of transmission within human populations, and the timing aligned perfectly with the timing of the Hajj. And so my colleagues at Johns Hopkins were losing their minds because they thought, this is it. This is the next pandemic is happening now. But it didn't. It didn't happen. And so I think this gives us another way of thinking about this problem and thinking about, what does it take for one of these pathogens existing within zoonotic reservoirs to spill over and cause problems. And in particular, I think the example of MERS-CoV allows us to think sort of deeply about how important is the zoonotic host. What does it matter where these pathogens are coming from? And you can sort of conceptualize this, as again is work by Benny Rice, by thinking about, say, for example, a mutational space, an arbitrary distribution of mutational space, and how that turns into pathogen fits. And if you, in the best case scenario, there is absolutely no fitness in humans. But if there is a capacity for this virus to replicate and spread within human populations, you would hope that a vast fitness valley separated what it takes for the virus to replicate in its reservoir and what it takes for it to replicate in us. Of course, that might not be the case. It might be that there is overlap. It can replicate a little bit within our population, so that red hump, as well as in ways that overlap with the set of mutations that exist in the reservoir. But it might be that fitness is below 1, so it can't actually take hold within human populations. This seems to be perhaps what's happening in the Middle Eastern respiratory syndrome situation, right? It doesn't seem to be able to take off in us, although it's trying all the time, and it does a couple of chains of transmission. And then, of course, something that we've often suspected, and we suspect, I think, also within the SARS-CoV-2 situation, is that there is some intermediate species, right? So it does great in pangolins or whatever else, and then it moves through a bat, and suddenly you have something that can transmit well within human populations. So all of these pieces will be determined by what that landscape of fitness and mutations looks like within that reservoir host and what it looks like within us. And I think there are pieces where we can really begin to pick apart what the mechanisms are. And one of my favorites in thinking about the importance of the zoonotic host is this question of the CpG ratio. So this is Cs followed by Gs in the genome. And it turns out that we have very low CpG ratios. And that's because Cs followed by Gs within our genomes are heavily methylated. And this, comes into play in all sorts of different ways. So we just don't have a lot of it. So when a virus spills over into human populations, one of the absolute sort of classic innate immunity line of defense is you look at little bits of DNA or RNA that are floating around. And if that thing has a high CpG ratio, that's a baddie, right? Because that's not us. So toll-like receptor 9 for its fans is something that goes looking for that, for example, one of the classic innate immune receptors. And it turns out if you look at so CpG suppression or CpG frequency, these things are correlated, and you look at SARS-CoV-2 over on the left, and you contrast it with MERS-CoV. SARS-CoV-2 seems to have come into our populations with a very low level of CpG. So it was insta-camouflaged. It was able to sneak under our innate immune system's triggers because it just somehow came in lucky. It seems to have been pre-adapted in some sense. Whereas MERS-CoV was not. So our innate immune system should be able to see that super quick and take it out. So, you know, that is one way in which emergent pathogens may or may not be successful, but also ways in which the zoonotic host reservoir matters. It was suggested that there was something about perhaps the bats that SARS-CoV-2 circulated in that would drove selection for quite surprisingly low levels of this CpG ratio. And sort of, you know, that ecology then becomes very interesting. That's innate immunity. There's also regularly spilling over into human populations might actually not be a great thing. So there was work in abattoirs in Niger, looking at people who worked a lot with dromedary camels. And what they found was that these people had T-cell immunity to this pathogen. So people who were, in some sense, regularly exposed to this pathogen were creating a sort of a shield of immunity that might protect the rest of us from it. They generated an immunity to it that way. So they never had symptoms. So possibly, you know, they were having mild infections and they were developing immunity that would prevent new infections and created some sort of barrier to it. This is something I mentioned in the case of the why are there only 4 coronaviruses. The receptor that Middle Eastern respiratory coronavirus uses is called DPP4. And this is a receptor which, for reasons that are mysterious to me, is at very high, apparently, or higher density in the upper respiratory tract in camels. And this makes it great for camel transmission, right? It comes into the upper respiratory tract, it easily moves into the next camel, but it's low in our lungs. And it's curious to me as to why that's the case, given that we're both mammals, but this is the fact, apparently. And this means that it is extraordinarily hard for MERS-CoV not to kill us, because as it starts replicating, it moves deep into the lungs, and if it's deep in the lungs, single cell will thick, you're dead if anything goes wrong, right? And so it just might be very hard for MERS-CoV to not be extremely deadly to us. And if it's extremely deadly to us, it will be very hard for it to move through our populations. Of course, you know, another sort of way in which it's important to think about this is that because it has such severe symptoms, we notice MERS-CoV spillovers a lot more than we might notice other spillovers, right? Like we have a massive selection bias issue here. It might be that all sorts of other coronaviruses are doing this all the time, but MERS-CoV does it in this dramatic way. So this might not be the perfect example for understanding what creates barriers. But I think it does give an interesting sort of at least conceptual map for thinking about the ways in which that host reservoir might matter and thinking about the parts of our biology that might constrain it or not in the context of what the zoonotic host is. But once it's moved from that host into us, then you have a kind of next series of questions, which is, you know, if it's in us, are there other things that stop it from turning into a raging pandemic virus? And there's really, I think, increasingly detailed mechanistic understanding of these various different pieces. There was some lovely work out of Alan Perelson and colleagues' group looking at the relative within host dynamics of Middle Eastern respiratory coronavirus in blue, and then the first SARS in green, and the most recent one in red. And what you can see is that the Middle Eastern respiratory syndrome guy just comes up super slowly and goes down really slowly. And it's, you know, probably there's lots of things that underlie that. The fact that it can't seem to duck under innate immunity quite as effectively might be part of it. We don't know. Maybe that cellular tropism is less effective. Like, you know, there's work going on various pieces of that. But if you're moving more slowly, you're less likely to get into the next person for obvious reasons, but you're also giving more time for these symptoms to catch up, right? Our symptoms often are our innate immune system getting into the mix as well. And if there's overlap between transmission and symptoms, there's a very classic paper from 2005 or something that made the point that what makes an infectious disease outbreak controllable, one of the key elements, is the degree to which symptoms and transmission are happening at the same time. If transmission is happening before you have any symptoms, that means you have asymptomatic transmission, you have invisible transmission, you have something you can't contain. The fact that SARS-CoV-1, as it was not called at the time, did not rampage around the globe is thought to be at least in part because it was possible to contain it, because every time someone was at risk of transmitting it, they were also very sick. So you could find them, you could contain it, you would know what was going on. I mentioned this already, but like if it just can't adapt to spreading with, one of the ways in which it may really struggle to adapt to spreading within humans is the location of this receptor that it uses. And this is not to say that cellular tropism doesn't change, right? It's not to say that the receptor that these different coronaviruses use can't change. There's an example of a pig coronavirus that started off as a mild respiratory infection in pigs, and then switched its receptor and became this very severe enteric infection. So, like all bets are off a little bit, but at least there's probably a somewhat unlikely event to happen. And so then the final piece is, do characteristics, so, we've talked about the zoonotic host, are within host biology, and then there's transmission from one host to another, so within our populations. A core result here, which I think is so fascinating and so weird, is that although millions of viruses are made within each host, right, I gave you that 10 to the 16 or whatever number it is, only like 2 to 8 make it into the next person. Right, so the kind of fitness troughs are extraordinary. It's extremely difficult for the right, if there's 2 to the 18 or whatever within me, one of them might be highly adapted, but the chances of it being in that 2 to 8 sort of life raft that's making it to the next host are really very low. So the bottlenecks are extraordinary. And this is likely to slow down adaptation in all of these viruses, but also in MERS-CoV. Yeah, it's also possibly the case that, that it's not, these things have a sort of hierarchy of transmission, right? They have to spread from cell to cell within us, and then they have to spread between us. And it's possible that the adaptations that allow you to spread from cell to cell within a host are the same as those that allow you to spread from me to another person. And it seems like that is possibly what happened for SARS-CoV-2, right? Like the mutations that made it able to stick to the ACE2 receptor better also seem to make it spread well within the host, but also between hosts. There's no reason for that to be true, right? There might be things that make you really effective at spreading within a host, you get to really high levels, and then that immediately blocks you from moving into the next host. We don't know, I think we don't have a really good sense of what the kind of case law is there of particular circumstances and particular patterns. And then, of course, very high virulence is just never a great idea if you're a pathogen, right? Like one of the reasons that SARS-CoV-2 was so devastating was it combined asymptomatic transmission with very severe outcomes in some individuals. If you're always killing people very quickly, you don't get very far if you're a virus. This is the homage to Jamie slide. So SARS-CoV-2 and MERS-CoV, both of them are known to have sort of super spreading characteristics, meaning that very few individuals are responsible for onward transmission, right? Many, many individuals are infected who never infect a single other person. This results in, this actually means that although outbreaks can be much more explosive, they're very likely to go extinct. Because just by chance, most of the people they infect won't go on to infect anyone else. And although SARS-CoV-2 and MERS-CoV share this characteristic, SARS-CoV-2 probably, it has a slightly higher overall transmission, so it can overcome that disadvantage, whereas MERS-CoV with its lower transmission is probably sitting in this part of the fitness landscape where it just goes extinct all the time. So I think when people talk about pandemic preparedness, they often talk about, oh, the index case or, oh, the critical mutation. But I think, thinking about both the Y4, but also the example of MERS-CoV, where we're beginning to sort of pick apart these different mechanisms, really suggests that, you need to think about the holistic bunch of stuff, as it's formally called. So, and of course, I mean, everyone in the room who's an evolutionary ecologist thinks about the fact that we would love a genotype to phenotype map, and we'd like that to also go to fitness. But that is something that is probably any efforts that narrow in on a singular focus are likely doomed to fail. Yeah. So a little bit on stepping back and thinking about this landscape, this evolutionary ecology of coronaviruses, I think was at least illuminating to me in thinking about how these emergence events might happen. I hope one of the things that came through in that was that the landscapes of immunity are kind of important, right? So and we know so little about it, right? We don't know whether there's a constant rain of coronaviruses onto us all the time, but very few of them manage to get through, or whether it's incredibly rare that one of us encounters a new coronavirus and it spreads within the population. We don't know whether pre-adaptation, so getting the right combination of mutations in the reservoir host, is the key bit, or whether it's, and then, that once they do that, then they're great, they're good to go, or whether it's meeting a human in the right way, that's a good way to go. And so to ensure that I never get Gates funding again, put up this incredibly funny slide where Bill Gates tweeted why I would rather encounter a shark in the wild than a mosquito, which is nonsensical, right? This is conditional on, that is conditional on the encounter, and this is not, right? We'd all rather meet a mosquito than a shark. I think we can agree. And I think this is one of the things that is just fantastic tweet. One of the things that knowing, so having a sense of whether any of us in this room showed signs of being exposed to coronaviruses or not, would tell us whether we're talking about sharks that we encounter very rarely, or mosquitoes that we're encountering all the time, but very rarely, in rare instances become really problematic. And that might inform how we proceed. I mean, I do think, for the record, that mosquitoes are much more likely, but it probably somewhere between the two. But it would inform how we proceed thinking about these pathogens, these emergent pathogens, right? If it's a shark, we want to think as hard as we possibly can about what makes a shark. If it's more of a mosquito, it's much more of a thinking about what are the exceptional circumstances that make this thing that can, sorry, the other way around. I'll just move on. I'm getting a little tired. So to sort of illustrate this, I'm going to shift away from emergent pathogens to think about a very common scourge, another piece of bad news wrapped in a protein, but this time the influenza virus. And this is a pathogen that is often emergent within human populations, but is also a regular endemic pathogen that circulates amongst us. And one, I think, that really illustrates the power of thinking about these landscapes of immunity. So the influenza virus, very bad news indeed. 36 million symptomatic illnesses in the United States. It's estimated, maybe 90,000 deaths. This is in a year. But the news is, of course, very variably bad across the life course, right? It kills you much more when you're older. And the ways in which this plays out is highly, highly different. And the things that shape these differences are multiple. So the first piece is that viral evolution. These viruses have hemagglutinin and neuraminidase receptors on their surfaces that our immune system can learn to recognize. And as our immune system learns to recognize them, they kind of run away. They change their shape a little bit. And so you end up with this kind of evolutionary arms race where our immune system is learning, the virus is running away. So you have to account for viral evolution if you're thinking about my risk to what viruses are circulating right now. You also have to think about immune dynamics, right? So have I been exposed to a similar looking virus in recent times, in which case my immune system might have learned how to deal with it? You also have to account for the fact that I might be senescing because my immune system might be falling apart. So that will also shape my risk at particular ages. And then, of course, you have to think about the fact that my risk depends on what all of you are doing, and if everyone got their flu shot, etc., or has been exposed recently. So there's many different pieces to unpacking how our landscape of risk to influenza shakes out across the life course, but much of this is rooted in how we think about how our immunity to influenza shakes out across the life course. And unlike, pathogens that are emerging in an erratic fashion, we have a sense of what this landscape looks like because we have huge amounts of data collection on this topic. And so, for example, what I'm going to show you now is some data that was collected in Guangzhou province in China. And what I'm showing you is the different strains of the virus on the x-axis, ordered by year of circulation. So this is the virus that was circulating in 68, and at the end, the one that was circulating in 2014. And then this is a vast cross-sectional study where they went to people of all these different ages, and they took their blood and they saw to what degree did their immune system respond to that particular virus, that circulating virus. And red means a bigger circulation, a bigger sort of response. And so, for example, If you take the virus that was circulating in 1968, and I think this data is from like 2014, then a bunch of people were too young to have ever seen that virus. So they had very low titers, but above a certain age, people are spiking titers. Their immune system recognizes that virus. And if you fill in the rest of this graph, it looks like this. So these are the super young people, and these are the responses to all the different strains. And if you squint at this, and if you're someone who loves thinking about nonlinear dynamics, you might instantly see a hint of multi-annual cycles bubbling up there. So there has been a vast amount of work on this sort of data. And we know, for example, that it seems like your immune system is completely obsessed with the first influenza strain you saw, right? So the thing you saw when you were 7, your immune system is like, that's the one. You'll see a later in life one, it'll still respond as if you were seeing that first one. And so it won't necessarily have the perfect shape to match the new one for a variety of reasons. And if we just to put it in cartoon form, if you have your first influenza infection, your antibodies might build up. They wane after a while, and they wane in titers because your immune system kind of forgets. They also are going to wane in effectiveness because the virus is going to change. If you get a second infection, there'll be some back boosting. So the things from before are going to come spiking up, but some new thing is also going to come into play. There might be a failed infection. You might have enough of a wall of antibodies to keep the new infection out. And you might get a new virus, a new pattern. We have a really good sense of the different rules. Well, we have a really good sense. We have at least a working model of how the different rules that shape this landscape, the mechanisms underpinning how we build up these walls of antibodies against the virus occur. And the way it works is that we know that there is boosting across different timescales. So a virus that looks a lot like the one I saw last year, those titers are both going to come up. If it's something I saw a decade ago, it's going to come up less. And it'll depend on how similar those viruses are. And we know that there is suppression. So the thing I saw when I was 7 dominates my response and kind of pushes down responses to later viruses for a very long time. We know that there's waning, and we know that there's waning in effectiveness as time kind of passes on. And we also know that there is going to be different patterns of infection risk over the life course. And if we put all these different pieces in, we can build a model that creates a pretty good prediction of that landscape of titers across ages and different strains. But it also captures the fact that within this data, there is a strong suggestion of a 20-year periodicity, right? So it's like we're all a sea in which there are these waves of antibodies circulating. And the ways in which this intersects with how our immune system is chasing the virus across the landscape, how our immune system forgets and boosted, results in this amazing 20-year cycle, which I think is just very cool. But it's not just very cool. I mean, it also matters because if we can put these mechanisms together to think about this deeply, one thing we know we can do is push antibody titers around. And we can push antibody titers around. We do it by vaccination, and we do it because of what it does to protect people from infection. There's a large question here, which is like protecting people from infection. How does that translate? How do these antibody titers also translate into thinking about changes in morbidity and mortality more generally, right? It behooves us to really unpack these various mechanisms because we can change them, right? And think deeply about how we change them. So the question then becomes, how do influenza antibody titers translate into changes in morbidity and mortality? And again, I was lucky enough to work with collaborators working with the Guangzhou Biobank cohort study. And this is a nested case control study where they had people who were sex and age matched and who were both deceased and living individuals, right? And we had their antibody titers to all these different strains. And so what I did was something very simple, and this was a pilot study, so it wasn't powered for significance, but I looked at the year of circulation of the virus strains, and I looked at the log odds of an increase in mortality. So if you're above the line, that means if the antibodies increase, you're more likely to die. If you're below the line, you're more likely to survive. And this is what the result looks like. Again, vast, not powered for this particular study, but really striking, right? There's a decrease in mortality titers of around 20%, which is stunning for a log increase in antibody titers. And it's stunning because we don't think that 20% of older individuals have flu, right? There's just not that much flu around. So it's kind of instantly intriguing, like what is the direction of causality? Is it just that people who are overall healthier have higher titers? Or is there something else going on? Is it a correlate of more general protection? So super intriguing as a pilot, but more intriguing was to come, which was that if you break this down by the sexes, what you find is that in males, you get an increase in survival with an increase in antibody titer, whereas in females, you get, possibly a reduction. So possibly higher antibody titers if you're female are associated with increased morbidity and mortality. And why might this be? I mean, it might be that there's not an increase, but it's just there's no pattern. There's a very vast confidence intervals. And that could just be because we know that females survive more than males. It could be this nonlinear averaging pattern where you're on the part of the curve where you can see an increase in males and you just couldn't see much in females. It might be that the female immune system brings a lot more weapons to the war. This is also known to be the case. There are very different actors in play. But it could also be that there are genuinely different patterns of how antibody titers shape survival, and at least in these older individuals in this particular circumstances. We don't know, right? There's no causal pattern here yet. It's all association and it's not powered, but it's super intriguing. And I was super intrigued and went digging into this and started reading more about sex differences in immunity. And the first thing you learn when you do that is that we know very little about sex differences in immunity. Here, focusing on sex rather than gender. But in 1977, as Sabra Klein pointed out in this lovely review, the Food and Drug Administration declared that you shouldn't do a clinical trial. They recommended that you didn't do a clinical trial on a woman of childbearing potential. So all women, pretty much. And then in 1997, they thought, maybe women are human. And then even in 2005, 8 out of 10 drugs that were withdrawn from the market were withdrawn because of side effects in women. So we just, we just don't know very much still. But there's really fascinating, so on top of this weird social cultural maelstrom of how the research to gender works, there's really interesting indications that there are very strong differences. So for example, 80% of autoimmunity is concentrated in women. And that's surprising. I mean, it seems very unfair. But it's also surprising if you're an evolutionary biologist. So evolutionary biologists are like, well, males, they invest in silly things. And therefore they are the weaker sex. So fabulous displays, weapons for competition, they take resources away from immunity, and so you end up with a situation. But that doesn't really explain it, right? That doesn't explain why you would specifically get higher autoimmunity, which is a very particular flavor of immune function, being increased in females. And in fact, if you think about it, so autoimmunity suggests that discrimination between self and non-self has broken down, and females are the sex that kind of carries the pathogen to term, and thus you would, if anything, expect greater tolerance, which doesn't seem to be the case. So I became really intrigued by this, and just to also prove to you that I am an evolutionary biologist, I built a really simple model thinking about how this might work, and you can lay it out as a very simple discrimination problem, right? So if you imagine that the blue bars are self and the red bars are non-self, and that your immune system is trying to decide where to draw the line that separates self from non-self. If you think that females are more autoimmune, maybe they're drawing the line further to the left, so they have more false positives, right? They're reacting to more of the blue range. And if you think about males in terms of their magnitude of response, it does seem like males have, this is my cartoon of how it works, there's a lot of complexity under the hood. But for example, septic shock is more prevalent in males. They have more natural killer cells. There's a greater CD8 to CD4 ratio. Just don't worry about the details. It's not inconsistent with males having larger numbers, bringing larger numbers of immune effectors to the battle, which would reduce your mortality associated with infection, but might also, be associated with various levels of immunopathology. So you can put this together in a relatively simple theoretical model to ask what the different investment in these two components of immune system function might be, and you end up with a fitness landscape that looks like this. I'm very happy to talk more about this with people who are interested in it, but the basic premise is that there's one group where you trigger the immune system even at very low levels, but you don't do a huge response, and another group where you trigger only at very high levels of like baddies being there, but then you do a massive response. It's consistent with those two patterns I'd observed, but not the answer, because there's no reason that females should be one peak versus the other. So I built out this model to put in a little bit more about what we know about how males and females operate, a little bit more life history, a little bit more, reproducing and such things, a little bit of investment in things like peacock tails in the males. Nothing could put the females on the peak I expected them to be on, where autoimmunity was a higher risk. So kind of, it's a very simple model, but intriguingly perplexing. And the only thing that really made it work was if you introduced the role of maternal antibodies, right? And this is where, if I'm infected, for example, by measles, any child I have will be protected for the first years of its life by antibodies that I transfer to it. And survival during that first year of life could be super important in fitness terms. And so greater autoimmunity might be the price you pay for having the sort of immune system that builds up a huge library of responses that you can pass on to your kid that will keep it safe for that first year of life. I think the way to go with this is that seahorses, the only known example of evolution of male pregnancy, the males pass antibodies onto their kids, and they do seem to have different immune systems in the direction you'd expect. And they have a whole spectrum of fantastic life histories that you could use to sort of tease this apart. So, you know, sex differences, vastly understudied, really fascinating. I think maternal immunity might be really important, but it obviously, you know, it obviously matters in thinking. It matters because it's interesting, but it matters also because how we deploy our various therapies is possibly made more effective by having more information on how this pattern plays out. To bring it back to emergent pathogens, you will, of course, all remember that age was one of the biggest drivers of, for example, hospitalization or death, but there were also really intriguing sex differences. And the common narrative was that males were dying more, but they were variably dying more across the life course. So we put this in the context of many different other sort of causes of mortality, just to see whether it was a particularly unusual phenomenon. And this is showing age. And here I was just looking at the relative risk of hospitalization. So if you're above the line, it's higher in males and below higher in females. And you get this very consistent pattern both for all-cause mortality, SARS and influenza, where before puberty, males have a much higher risk of hospitalization. At later ages, males are higher, but between the two, for at least the infections, you end up with a greater risk in females. You can sort of think of this as being males as the weaker sex, but this is this role of danger of immunity, which, you know, as we think about how we deploy therapeutics, for example, one of the key weapons we brought to bear on the SARS-CoV-2 pandemic was shutting down your immune system, because that was what was killing people quite a lot. So drugs like dexamethasone were effectively steroids. Something that I'm super excited about learning more about this landscape of immunity. I think if I could go out and take all of your blood and figure out what you've been exposed to, that would be really creepy, but kind of fascinating. And we've written a lot of papers making arguments for the value of a global immunological observatory in this landscape. But with that, I just want to thank so Benjamin Rice led most of this work. Rachel Baker, a vast review we wrote. Bing Yi, who's now a professor at Hong Kong University, did that work on influenza I talked about, and then there's a couple of others, and thank you very much.
Thank you, Jess. Dan.
What's going on sex-specific autoimmune stuff in birds?
So that's fascinating, right? Because birds are, so sex differences are both going to be rooted in chromosomes and in hormones, and birds are great because they have the reverse sex determination system. I don't know very much about autoimmunity in birds, but I think it'd be wonderful to find out more. There is, for example, so just to not talk about birds because I don't know much about birds, but there is many immune genes sit on the X chromosome for reasons I don't understand. And one of the toll-like receptors, this is one of the innate immune receptors, sits on the part of the X that isn't silenced, right? And so it has been associated with both increased autoimmunity, but also increased protection from influenza. So it's I think it's fascinating, and I'd love to know more about how why would immune genes be sitting on the X and how it might differ across species.
Yeah, great talk. I have a question about I guess, controls on diversity in any pathogen diversity in any organism. I was wondering if you could expand on what you were saying about the small number of endemic coronaviruses to make a guess as to why there are so many within pigs or within humans. I don't know what the most diverse group of pathogens are, but why is there a high risk of that pathogen not coronavirus?
I wish I had a general theory of pathogen coexistence and diversity for you. There are so many, so we've spent a lot of time spinning our wheels around this, and they're really interesting pieces. Like, you know, it seems like if you're a respiratory infection, your diversity is possibly lower, possibly, who knows exactly why, but things like gut viruses, the enteroviruses live in gut, hundreds of species. And that's, you know, maybe like the gut is just a different environment, right? Like we're coexisting with a bunch of different microbes there. Like it's not like the immune system, which is super precious and super defended. There's also a lot about the degree to which, you know, there's a piece about the habitat they're living in, but there's a piece about the immune system and the ways in which immune memory is developing. There's been an argument that it's actually very hard to develop lasting immunity to things within our upper respiratory tracts because there's just so many of them and so much exposure. This is Jonathan Udell's paper. But how does that translate into diversity? Is it, you know, I just don't know. I think it's a wonderful area for speculation, but no science yet.
Yeah, do most autoimmune diseases happen post-reproductive age in females, and is there therefore like weaker selection pressure to find that?
Great question. So there are like, you know, there's kind of, if you look at the things that are formally classified as autoimmunity, there's not that many of them. There's like 16 or something, and a few of them are male specific, and many of them are found in males and females, but overwhelmingly in females. There are one or two that are found in younger individuals, but most of them are after maturity. I think that's a really great question, right? What is the strength of selection on them if they're happening later? One of the things I find really fascinating is that the genetic underpinnings of many of these autoimmune diseases are the same and show signatures of positive selection, suggesting that they were super important in defense against a pathogen back in the past or something like this. But I mean, I think there's a lot to be unpacked there in terms of thinking about what that age course looks like and how it translates into persistence over the long term.
So I thought it was really interesting that in answer to Dan's question about birds, you immediately went to XY versus ZW. Where I go with it, I said reptiles and amphibians is live bearing versus
Right.
and you know having a fetus, which of course birds do, and animals are virtually almost live bearing. So I guess I'm curious, why did you go there? To me, it would have made so much sense with the autoimmune thing to say, well, you've got this other thing, you know, nurtured inside of you, and you've got to have your immune system has to deal with barrier, and in which case, look at chickens, look at lizards, look at xanthus, look at whatever to see what's going on there.
Oh, that's a great thought. I wonder if it's partly because the model I made had no space in it for pregnancy. But I also, I mean, I think it's a fascinating. So the seahorses I'm obsessed with, there's a sequence of life histories within the cygnathids, which go from like the females dropping the eggs to the ground, the females gluing the eggs to the males' tails, the males having kind of a pocket that the females stuff the eggs in, and the males having a full-on pouch that does gas exchange and transfers antibodies. So I think that would be a really fascinating way to start unpicking this. But I mean, I love a reptile even more, so. Yeah.
Has there been any look at like they're looking at the viruses as a community, and so it is potentially like they're talking about maybe individual viruses, but if you are co-infected with something like say influenza virus or maybe some other bacteria, if that could leave you vulnerable to greater chances of presenting these cases, or in that case, presenting infection.
So that's a great question, and there's a wonderful layering of ways in which this can happen. Obviously, HIV, which takes out your immune system, makes you more vulnerable to a bunch of other stuff. So that's a sort of natural co-infection piece. There are ways in which, so I think there's a bacteria that slices off a particular receptor that makes cells more accessible to a particular, I forget the exact story, but there's certainly places where there are known to be pathogens that make the environment more conducive to further invasion just by changing the landscape. Yeah, so there's a whole spectrum. And then of course, you know, there's the if you've had any sort of like a rhinovirus infection, your innate immunity tends to come up, and that might make you more protected against others. So I think that the direction of the kind of valency of this relationship can go in all sorts of different ways that will probably depend on the specific pathogen. Yeah, the microbiome is of course an obvious way to go in thinking about that, and people often think about the ways in which your microbiome I keep going in the protection direction instead of the facilitation direction, but basic point is yes, lots of different interesting examples. Yeah.
I was wondering if there's any correlation or any relationship between the severity of the virus and the transmissibility, and if the reservoir is a domesticated or wild species. Because I guess thinking about the camels and pigs and even with like cowpox and stuff, there's certain groups of people who like small groups who are in regular contact with the domesticated animals, and so they have built up an immunity versus like animals that are caught in the wild for food, where people don't really have that much contact with them unless they're dead or about to like they just don't have as much regular contact with like a bush meat type. Is there any relationship between illness severity and reservoir?
You know, that's really interesting. I don't actually, I wonder if Jamie knows a better answer.
Broadly, like people hypothesize about pathogenic distance and the alienness. Some people have argued that that'll make it more likely to be severe. I think it seems like it would just be more random, random virus space. But there is a correlation with bats in particular. Right here, we're talking about bat immunity and the importance of innate immunity shaping the viruses in the zoonotic pool. And there's a theory, like Carol Brook in Chicago now, about how bats have intrinsically scaled up innate immunity, and it trains their viruses to be really aggressive. And then when they hit us with their wimpy, down-regulated innate immunity, they ruin us. That's a strong working theory at the moment.
To what extent are you able to combine all these criteria for successful outbreaks to kind of create like a watch list of problematic viruses?
I think that's what Jamie did. No, I think so. I think there's always work to be done, right? I mean, I think that I would be fascinated. I think the more we think about these things, the more things pop up into our minds. Like I'd love to know an atlas of all the receptors that are in our lungs, right? Like that seems like a really fascinating question that would be kind of low-hanging fruit. And having a sense of what receptors viruses are using out there, you know, it's not going to be one answer. There's going to be a suite of things that we can think about in the space, and Jamie has thought about it much more than I have. Yeah.
Going back to the X-Y spiders or sex differences in species that are hermaphrodites.
No, I'd love to. Wouldn't that be fascinating? And also, like, you know, the fish that change sex. So it'd just be amazing. Yeah, no, but no, sadly, no. Hi.
Yeah, so explains the basic microbiome and more about the page and about.
Oh, cool.
I'm sort of curious whether what the rate would estimate, not just in coronaviruses, but kind of across the whole viral tree, sort of like neutral pandemics, if that makes sense, or at least pandemics that don't have a like large effect on health, and whether any differences that might be observed there in the coronaviruses, maybe some phylogenetic conservation of some, you know, like implicit like bad thing that coronaviruses do. Does that make sense? So basically, like, are there lots of neutral pandemics happening?
I'd love to know. I mean, maybe Jamie might know better than I do, but I mean, I think this is one of the reasons I'd love to have a sense of what the landscape immunity is, like what is normal. One of our friends and colleagues, Saki Takahashi, does a lot of sampling in Uganda, and we were talking about, it's kind of mysterious, there isn't vast burden of dengue in Africa. And she was like, you know, it's funny, like there's this flavivirus that everyone seems to be immune to that is asymptomatic, so we just don't care. And I was like, really? so I don't know, but I think we will know. Hopefully we will know as these sorts of data start coming in. And, you know, I think that it would be sensible in that situation to look for the immune signature rather than the pathogen itself, because the immune signature is this window onto the past, right? So you can, whereas the pathogens might be, you know, there and gone really quickly.
May I use my prerogative up here for 1 minute? So wonderful talk, loved it. The premise of the first part of your talk was why are there only 4 endemic coronaviruses? I think debatably there are now 5.
Oh, yes, true.
And so I'm curious whether you think SARS-CoV-2 is going to squeeze one out, or has the space expanded somehow?
I'd love to know that. I mean, what do you think?
You get free lunch. Yeah, fascinating. Yeah, thanks very much again. Come back next week. We have a very amazing speaker next week, our own Nandita.